Hello, thank you for watching these poster presentations. So my name is Jie Hou from the Computer Science Department at St. Louis University. Here we are presenting our recent work in the protein structure analysis that aims to rank the protein-protein interaction structure using the deep graph neural network. It is well known that the accurate protein structure prediction is crucial to help us understand the protein functions in our living cell, in the living cells. So the recent advance in the single-chain protein structure prediction brought the great promise in the computational algorithms to the protein-protein interaction. Therefore, we propose the effective protein model ranking methods would be quite useful for the structure prediction approach to select the most accurate protein structure and further improve their model prediction accuracies. So the study applies the deep graph neural network to score the protein complex model by utilizing the residue level structural information in 3D space and the sequence level co-evolutionary constraints. Because our model treats the protein structure as a connected graph in which each node represents the residues and each edge represents the closeness between any pair of residues in the complex structure. So therefore, the graph network is well suited in these problems to effectively integrate the spatial information for 3D structure quality learnings. So in our study, we designed the several features for the structural quality learning, including the node features and the edge features for the graph neural network. So specifically for the graph nodes, we're going to use the physical and the chemical properties of amino acids and per residue energies in the structures. And for graph edge, we use the inter-residue distance and the co-evolution constraints from sequence alignments. We can talk about this one in the later feature sections. Overall, the algorithm aims to provide the residue level estimation of the model quality accuracy in terms of the interchain and the interchain LDTT scores. So to train our deep learning architectures, we utilize two different datasets. The first dataset is a single chain protein decoy datasets comprised of almost up to around the 8,000 proteins, and as well as up around the 100,000 protein structure decoys to training the model to give us the residue specific quality assessment for single chain proteins. In order to evaluate the interface residue accuracies, we also incorporate the complex targets from the ZDoc datasets that has around 176 complex targets, and each target comprised of the 10 of thousands predicted decoys made by the ZDoc. So in our study, we use the top 501 to up to 1,000 top ranked predictions to training the deep learning model to give us a prediction for the interface residues. Okay, so in order to generate features, we utilize the sequence features and the structural features. For the sequence features, we first generate the multiple sequence alignments for every individual sequence chains in either single chain proteins or complex proteins. For single chains, we can generate the interresidue, predicted interresidue distance, predicted segment structures, or evolutionary conservation features. These sequence features gonna be encoded the node features combined with the structural features we can derive from the 3D models, including a list of these structural features to describe the local quality of every amino acid in the 3D models. In order to encode the edge features, we also can extract the observed interresidue distance from 3D model, combined with the predicted interresidue distance to encode the edge embedding for the graph learning. So here is the architectures we show in our models. Input is the 3D models, either single chain proteins or complex proteins. So we extract the residue specific features and residue pair features from the 3D model and predict the two major types of outputs. So if you input the single chain proteins, it will generate the inter-chain LDT score and give the prediction of the accuracy for every amino acid in the whole sequence. If the input is the protein complex model, it will also give us the quality prediction for a list of the interface residues. We define this interface residues score as the interchain LTT score. So these are the two types of the outputs, and both outputs can be combined to rank the protein, either single chain proteins or complex proteins for the computational approach. Specifically for the interchain LTT score, it's the common LTT score to describe how well the local interactions in the native structure is conserved, can be found in the 3D models. It's basically defined as the averaged fractions of the local interactions defined by 4 different cutoff. In addition to account for the interface radius accuracy, we define the interchain LDT, which is also average of the fraction of the local interactions. However, the local interaction defined by the local radial pairs is found from different protein chains in the complex model. For instance, every residues in one chain, you're going to looking for the distance pair in another chains and then derive the fraction of the local residue interactions that can be conserved in the 3D models. In our analysis, we found that the interchain LDT score is much better to account for the protein complex ranking compared to the interchain LDT score. So for instance, if we calculate the protein model complex models according to the interchain LDT score, you can see it's much easier to differentiate from the good models from the bad models. And that's why we actually introduced the interchain LDT score as the major output of the network to predict the quality of the interface residues. Overall, we just show our initial results from our current model trainings. We are still trying to improve the model training. Here we can show that we can, according to the ISMB loss, which is defined by the absolute difference between absolute difference of the ISMB between the top ranked model and the best model for every target. And we calculate the average ISMB loss over all the training targets and the task test targets, and compared to the ISMB loss by the Z-rank models. And we can observe a slightly better performance. And also showing, according to the results we're showing here, we can see that the predicted global LDT score by the interchain LDT is also very close to the actual LDT scores. And we're hoping our improved models can give the much better performance in the future work. So yeah, that's everything for our. This is very basic introduction to our recent work. So looking forward to chat with everyone in details in the conference. Thank you very much.